Hi, so the paper I've chosen is on the topic of anti-CRISPR proteins. More specifically, it talks about how anti-CRISPR associated proteins repress anti-CRISPR transcription and the importance and significance of the anti-CRISPR associated proteins' role in terms of the bacteriophage's survival. So here we have the title of the paper I've chosen, and then we have the bibliographic information. Okay, so some general background information. This is just some general background information from the paper. It kind of summarizes the vital information needed to understand the research and the studies that were conducted in the article. So first we have that bacteriophages express anti-CRISPR or ACR proteins to inhibit the CRISPR-Cas systems that would destroy their genomes. Most ACR genes are adjacent to anti-CRISPR associated genes. An anti-CRISPR associated promoter drives high levels of anti-CRISPR transcription immediately after phage DNA injection, so the initial step of the infection cycle, and ACE genes or anti-CRISPR associated proteins subsequently repress this transcription. Without this anti-CRISPR associated activity, the strong transcription of the anti-CRISPR proteins would be lethal to the phage. Sufficient levels, so notable levels of anti-CRISPR proteins accumulate early in the infection process in order to inhibit existing CRISPR-Cas complexes in the host cell. Lastly, the conserved role of ACE proteins is to alleviate the deleterious effects of strong transcription from anti-CRISPR promoters. Okay, now moving on to the first figure. We have this, and this is just like a graphical demonstration of the effect that anti-CRISPR associated proteins have on the phage's survival. So as you can see on the bottom left right here, the presence of anti-CRISPR associated proteins, the anti-CRISPR protein transcription is reduced, and so the phage, let me just show, the presence of the anti-CRISPR associated proteins allows for the anti-CRISPR protein transcription to be reduced. And as we know, high levels or strong levels of anti-CRISPR transcription is lethal to the phage. So this is actually good that there's only like one. And so with this, the phage survives. On the other hand, without the presence of ACE proteins, ACR proteins are not, ACR protein transcription is not inhibited, then anti-CRISPR transcription is not reduced, and so this obviously accumulates strong levels of anti-CRISPR proteins, which is lethal to the phage, and also it disrupts downstream gene expression, and the phage dies. So, yeah, moving on. Okay, here we have a schematic representation of the genomic context of diverse anti-CRISPR genes. So this is just, to show how diverse anti-CRISPR genes are. And so the colored arrows represent anti-CRISPR and anti-CRISPR associated genes, as well as helix, turn helix, HTH motif proteins. Genes in the same color are from the same family, and other genes are shown in gray, and the predicted functions are shown as well. Okay. So next we have this, and this is just data from one of the experiments done in the paper. So this kind of demonstrates how a loss of the anti-CRISPR repressor activity affects transcription of downstream genes. And so A through D are just showing the relative transcription levels of the phage from a wild type um, version of the phage and a mutant version of the phage without the anti-CRISPR associated gene. And so these were determined by QRT-PCR. So in A, the genes that were assayed was the ACE gene. In the B, the gene that was assayed was a transposase gene, or A. And then C is the gene assayed was the B gene, or the IZ gene, which is a scaffold gene, and this lies directly downstream of the anti-CRISPR associated gene. And D, we have the G gene, which is actually upstream of the of the ACA gene. So as you can see, the gene that was downstream of the ACE gene, which was graph C, its transcript levels in the wild type and mutant phages were very different. It shows that the mutant phages experience like a dramatically decreased level of transcription. And this coincides with the claim that I had earlier, how loss of anti-CRISPR associated activity um, leads to reduced transcription of downstream genes. Okay. Next, we have another graph, and it shows kind of how ACE genes, or how in particular this ACE gene, which is ACRL-F1, has its own promoter. And so A is just the transposase gene that we had in the earlier slide, and G is the late expressed gene. And so as you can see, there was like more than 100-fold increase in the level of anti-CRISPR transcript within the first 20 minutes. So that's the black bar, that's the ACE gene. And so as you can see in the other genes, how ACRL-F1 transcription was initiated earlier than the other genes. And A was actually, since it's an early expressed gene, it was actually expected to be one of the first genes transcribed. However, this is not the case, as is shown here, as the ACE gene was obviously transcribed before the A gene. Transcription of G, which is located upstream of the ACE gene, was expected to increase late in the infection process. And so it did, as you can see, that it didn't really reach notable levels until after about 50 minutes. And so this data kind of shows that the ACRL-F1 transcription occurs very early in the infection process, and this is presumably to allow the anti-CRISPR protein to accumulate to sufficient levels in order to inhibit the CRISPR-Cas complexes that are already present in the cell. And this kind of transcription profile also indicates that the ACRLF1 gene, or the ACE gene, it possesses its own promoter. Yeah. So these are kind of the main conclusions that are surmised from the paper, and it is that anti-CRISPR associated proteins function as repressors of anti-CRISPR transcription, and anti-CRISPR genes are transcribed to high levels quickly after phage infection, the ACRLF1 gene is transcribed from its own promoter at the onset of phage infection, and that anti-CRISPR-associated function removes deleterious effects of high anti-CRISPR transcription, which is, of course, beneficial to the phage. It allows it to survive. Okay, and these are just some future questions about how anti-CRISPR-associated proteins um, can maybe benefit or yeah. So one of them is being, how can ACE proteins be modified in order to be utilized in medicine? Another one is, how can the principle of alteration of transcription of the downstream genes be taken advantage of? And then finally, it's the identification slide with my name, my TA, and a picture of me. Thank you for listening. I hope you have a great day.